Welcome to today's section. Today I will be taking you face to face with one of the most dangerous emerging zoonotic viruses in the world, the Nipah virus. Nipah virus is not just another viral pathogen. It is a highly fatal, rapidly emerging zoonotic virus with the ability to cross species barriers, cause severe neurological and respiratory disease, and trigger outbreaks with devastating consequences for both human and animal health. First identified in 1998 during an outbreak in Malaysia. Nipah virus has since caused repeated outbreaks in South and Southeast Asia, particularly in India and Bangladesh, with case fatality rate reaching 40 to 75%. What makes Nipah virus especially alarming is its natural reservoir in fruit bats. Its multiple route of transmission, including animal to human and human to human spread, and the absence of a licensed vaccine or specific antiviral treatment to date. In this presentation, I will walk you through the epidemiology, transmission, clinical manifestation, diagnosis, prevention and control measures related to Nipah virus. Nipah virus is a deadly RNA virus belonging to the family Paramyxoviridae and the genus Nipah virus. In the Nipah virus, two type of viruses are there. One is Nipah virus, next is Hendra virus. Hendra virus mainly affecting horses and Nipah virus mainly pigs. Although it has emerged only in South and Southeast Asia, it has quickly earned attention as a highly fatal zoonotic disease with a case fatality rate exceeding 60%. Its natural hosts are the fruit bat of the genus Pteropus, which silently carry the virus, allowing it to spill over into other animals and humans. The virus first made headlines in 1998 in Malaysian state of Perak, where pig farm workers developed unusual severe encephalitis. This outbreak marked the beginning of global awareness of Nipah virus as a serious public health threat. In 1999, Nipah virus was formally identified as a paramyxovirus and found to be closely related to the Hendra virus. Human infections typically occur through a spillover of virus from fruit bats to domestic animals such as pigs when they act as an intermediate host, transmitting the virus to humans. This highlights the critical role of animal to human transmission in Nipah virus outbreaks and underscores the importance of monitoring both wildlife and livestock to prevent future cases. Etiology: Nipah virus is an enveloped RNA virus classified under the order Mononegaviridae, family Paramyxoviridae, and genus Nipah virus. The virus gets its name from the village of Sangai Nipah, where the first viral isolate was obtained. Being enveloped and lipophilic, Nipah virus is susceptible to common disinfectants, including 70% alcohol and soap, which makes hygiene and proper disinfection important in preventing its spread. About the history, the first major Nipah virus outbreak occurred in Malaysia between 1998 and 1999, causing severe encephalitis in humans. Over 250 people were infected with more than 100 deaths, highlighting the virus' highly fatal nature. In pigs, the outbreak caused respiratory and neurological syndrome, and the virus was successfully isolated in March 1999. To control the outbreak, around 1.1 million pigs were culled out of a total 2.4 million, and fortunately no new cases has been reported in Malaysia since 1999. Factors such as urbanization, deforestation and drought contribute to the spillover of virus from bats to the domestic animal, emphasizing the role of human-environmental interaction in emerging infectious diseases. After the initial outbreak in Malaysia, 1999, saw new cases in Singapore, where abattoir workers were infected after exposure to pig imported from Malaysia. In India, Nipah virus outbreak has occurred in 2001, 7 and 18. After 2018, most of the year there was regular report from India. And also in Bangladesh, outbreak has been reported almost every year since 2001. In 2014, a Nipah virus outbreak occurred in the Philippines affecting horses and humans. It was similar to the Malaysian strain linked to the consumption of undercooked horse meats. Interestingly, Nipah virus outbreaks show a seasonal pattern occurring predominantly between January to May, highlighting the importance of heightened surveillance during this period. This map provided by the CDC shows the distribution of the Pteropus fruit bat alongside with non-Nipah virus outbreak. As we can see, the regions with a dense Pteropus bat population often overlap with areas of human and animal infection. These blue dots are the Nipah virus infection and the red dots are the Hendra virus outbreaks, emphasizing the role of these bats as a natural reservoir. Understanding this distribution helps public health authorities identifying high-risk zones, plan surveillance, and implement preventive measures to reduce the risk of spillover events. Actually, if we look into the Bangladesh outbreaks, one of the primary route of Nipah virus transmission to humans is through the consumption of raw date palm sap contaminated by the infected fruit bats. Bats often lick and urinate in the sap, collecting pods, allowing the virus to enter the sap. Studies have shown that the Nipah virus can survive for up to 4 days in bat urine and for 1 day in this contaminated sap, which kept in a temperature of around 90 degrees Celsius. Nipah virus is present in various bat secretions and excretions, which highlighted the risk of direct and indirect exposure to humans and other domestic animals. This emphasized the importance of avoiding raw sap consumption and implementing measures to prevent the bat contamination of the food sources. In India, the Nipah virus outbreak has been reported in 2001 in Siliguru and 2007 in Nadia, and from 2018 onward from Kerala, and there was some outbreak in 2026 in West Bengal also. The genetic analysis revealed the virus in India are similar to the Bangladesh strain, highlighting cross-border virus circulation. Transmission in this outbreak was primarily human to human, including nosocomial transmission in hospital setting. That we have to remember in the case of Malaysian and Bangladesh strain. In the Malaysian strain, we can see an animal intermediate in between the bat and the humans. But in the case of Bangladesh and in the humans, there is no animal intermediate is there. It is direct bat to human and from human to other humans. Beyond India and Bangladesh, evidence of Nipah virus has also been found in fruit bats in Cambodia, Thailand and Madagascar, even in bats that show no clinical signs, suggesting a widespread natural reservoir in these areas. So I already told in the previous slide, like two transmission patterns are there based upon the strain. In the case of Malaysian strain of Nipah virus, primarily transmit from bat to pig and then to humans and other animals. And human to human transmission is rare in those cases, but has been documented in some cases, including some seroconversion in healthcare workers and instance of late onset Nipah virus infection. Genomic studies show that this strain is relatively homogeneous, indicating minimal genetic variation among virus strains. But in the case of Bangladesh and Indian strain of Nipah virus, it is primarily transmitted directly from bat to human, unlike the Malaysian strain. And human to human transmission is more common. And nosocomial transmission or spread within healthcare settings has also been reported, emphasizing the importance of strict infection control measures. Experimental studies in ferrets has been shown that these strains are shed in large amount in oral secretion, suggesting that saliva and respiratory droplet play a significant role in viral transmission. Genomic analysis reveals that these strains have 6 additional nucleotides compared to the Malaysian strains. Furthermore, the viruses are highly heterogeneous, indicating multiple independent introduction from bats into the human population. Understanding these transmission patterns and the genetic diversity is critical for outbreak management and the development of preventive strategies. A study by Yadav and colleagues in 2012 conducted in Maharashtra and West Bengal detected the Nipah virus RNA in Pteropus bats, suggesting that these bats may serve as a natural reservoir for the virus in India. Phylogenetic analysis of the Nipah virus sequence revealed two distinct lineages. One lineage includes sequences from the Bangladesh and India, while the other comprises sequences from Malaysia, Cambodia, highlighting the geographic clustering and genetic diversity of the virus. So there is two clear lineage, one that will be clustering between the Bangladesh and India. So its transmission pattern is different from that of the next lineage, that is a Malaysian lineage. In the Malaysian lineage, there is animal intermediate between the transmission, but in the case of Bangladesh and Indian lineages, here the transmission is directly from bat to the humans. About the epidemiology, the primarily natural reservoir of the Nipah virus is a fruit bat of the genus Pteropus, and domestic pig act as an amplifying host, facilitating transmission to humans, mainly in the case of Malaysian outbreaks. In addition, infection has been reported in several other animals, including dog, cats, horse, and goats, highlighting the virus' ability to cross species barriers. So the transmission in the Malaysian strain, initially from Pteropus bat, it is a reservoir of this virus. It is transmitted to the pig intermediate, from pigs to the other animals like horses, our pets like dogs and cats, and from dogs and cats there is a rare chance of transmission to human. Mainly transmission to the human occurs by pigs and the pig products. Actually, these cats are experimentally infected, and dogs, in the case, natural infections are reported, but through pig only. But in the case of Indian and Bangladesh strain, what we noticed was there is no intermediate animal or involved from the fruit bat. It is directly transmitted to the animals, and also from human to human transmission also there. That's why there is a regular high number of outbreaks are reported in these countries. About the source of infection, the Nipah virus can be found in urine, uterine fluids of wild Pteropus bats, and experimental studies have successfully isolated the virus from urine, kidney, and uterus of infected bats. Humans and other animals become infected through contact with fruits contaminated by bat saliva or urine, as well as through contaminated drinking water or exposure to aborted bat fetus and other reproductive issues of fluid. This was in the case of Bangladesh and the Indian strain. And the risk of transmission is further increased by the growing overlap between the bat habitat and piggeries. In domestic pigs, the virus is shed in respiratory secretion, saliva, and urine, which can serve as a source of infection for humans and other animals. This is in the case of the Malaysian strain. About the resistance to physical and chemical action, the Nipah virus is inactivated by heat, specifically at 60 degrees Celsius for 60 minutes, and the virus is stable over a wide pH range from 4 to 10. Common soaps, disinfectants, lipid solvents such as alcohol, ether, and sodium hypochlorite solution are effective due to the enveloped nature in inactivating the virus. Under favorable environmental conditions, the Nipah virus can survive for extended periods. It has been shown to persist for several days in fruit bat urine and in contaminated fruit juice, highlighting the potential for indirect transmission. About the Nipah virus pathogenesis, the entry of the Nipah virus into the host cell is mediated by the receptor efferin B2, a membrane-bound ligand expressed on endothelial cells and neurons. The virus is highly stable and possesses a non-segmented genome, contributing to its ability to infect a wide range of tissues and hosts. It is also known by names like porcine respiratory and neurological syndrome, porcine respiratory encephalitic syndrome, and barking pig syndrome. About the symptoms, the Nipah virus infection in pigs usually varies with age. Suckling piglets less than 1 month old often shows coughing, open mouth breathing, abnormal posturing, and convulsion. That is mainly some respiratory and neurological manifestation. Mortality in this age group can be high, reaching up to 40%. In young swine, that is aged from 1 to 6 month old, the disease primarily manifests as respiratory signs, including loud, non-productive cough, sometimes referred to as barking pig syndrome or 1 mile cough. The adult pig may exhibit both respiratory and neurological signs, as well as reproductive manifestation like abortions, although some affected adult may remain asymptomatic. Symptoms in dogs include it will have some distemper-like signs, fever, respiratory discharge, also show ocular and nasal discharge. In the case of cat, fever, depression and respiratory manifestation. In horse, usually it is encephalitis. In humans, the Nipah virus infection present with fever, headache, vomiting, myalgia and some cases respiratory symptoms such as emphysema. The infection can progress to encephalitis, which may relapse even after initial recovery, as well as meningitis, disorientation, persistent neurological deficits, coma and death. The case fatality rate ranging from 40 to 60%, reflecting the high pathogenicity of the virus and the need for prompt medical intervention and supportive care. About the lesion, the Nipah virus infection does not produce any pathognomonic lesion in the respiratory tract and co-infections are usually common. In the central nervous system, the meningeal edema with congestion can be observed and the pulmonary lesions include consolidation with petechial and ecchymotic hemorrhages and distended interlobular septa. About the microscopic lesion, this Nipah virus infection can lead to pneumonia characterized by the syncytial cells in the respiratory epithelium and endothelium also. Mononuclear vasculitis with fibrinoid necrosis is often observed and may be associated with the thrombosis, reflecting the virus vascular and the systemic impact. It also shows infiltration of alveolar macrophages and neutrophils in the lungs. In small number of pigs, CNS manifestations are observed, including non-suppurative meningitis. It's a feature of a viral infection. There is perivascular coughing and gliosis. It can be seen in most of the viral infections that is affecting the brain. So it's not that much specific to the Nipah virus. The virus can also be demonstrated in the meninges, confirming its neurotropic nature. About the differential diagnosis, in areas where Pteropus bats are present, any respiratory or neurological conditions in swine should prompt considered for Nipah virus differential. Specific signs among pig include death of suckling piglet, sudden death in boars or sows, abortions and other reproductive dysfunctions. Respiratory signs may present with harsh, non-productive coughing, while encephalitic manifestation can include trembling, muscular incoordination, and myoclonus, often progressing to lateral recumbency. Before collecting and sending any sample suspected of containing Nipah virus, proper authorities must be contacted. The sample should only be transported under secure condition and to authorized laboratories to prevent accidental spread of the disease. Appropriate samples for diagnosis include throat, nasal swabs, CSF, urine, blood and tissue samples. Strict biosafety precaution must be observed during collection, handling and transportation of these specimens. About the serological testing for Nipah virus include ELISA, which is safe and quick, and the serum neutralization test, which is considered the reference serological test. Immunohistochemistry can be performed on the formalin-fixed tissues such as lung, kidney, spleen, and heart. Viral antigen can be detected on the endothelium and the cellular debris within the lumen of the upper respiratory tract, as well as in the porcine meninges, lungs, trachea, and kidney. In pregnant animal, viral may also be found in the uterus, placenta and fetal tissues. It is important to note that these diagnostic tests do not have perfect sensitivity or specificity, and results should be interpreted in the context of clinical science and epidemiological informations. Virus isolation can be performed from tissues such as kidney, liver, spleen, tonsils and CSF. Nipah virus grow well in green monkey kidney or Vero cells and rabbit kidney RK13 cells, producing cytopathic effect within 2 to 3 days. Characteristic changes include large syncytia formation with nuclei located at the periphery of the cells. Confirmation of the isolate in the cell culture can be done using immunofluorescent testing or immunoperoxidase staining. Definitive diagnosis is made by virus neutralization test, including plague reduction, microtiter neutralization, and immune plague assays. Electron microscopy can be used to examine the cultured virus or lesions, revealing the characteristic nucleocapsid and the viral membrane typical of paramyxovirus. Molecular detection using PCR serves as a reliable diagnostic tool. Laboratory confirmation of Nipah virus infection can be achieved using serum neutralization, ELISA, and RT-PCR. Due to its high pathogenicity and potential for human-to-human transmission, Nipah virus is classified internationally as a biosafety level 4 agent, requiring highest level of containment in the laboratories. About the control measures, effective management of pig farm is critical to prevent the Nipah virus infection. In countries where Therapist bat acts as a natural reservoir, pig farm should be located away from the fruit bat and other vegetation that attract bats. Strict biosafety measures should be implemented to prevent the introduction of the infected animals. If introduction of a new pig is necessary, strict quarantine procedure must be followed. Farmers should avoid feeding pigs partially eaten fruits or vegetables which may be contaminated by these bats. In the event of animal death, burial site should be disinfected with chlorinated lime or sodium hypochlorite to prevent environmental contamination. Education and the proper use of personal protective equipments by individuals exposed to potentially infected pig is highly recommended. Improved hygiene at pig operation is essential to reduce the risk of transmission. Handlers should wear gloves and masks when dealing with infected or suspected animal. Hands, utensils and clothing should be thoroughly washed with soap solution after contact with pig or contaminated material. And in countries like India and Bangladesh, avoid consuming any partially eaten fruits that is found in the wild.